the liver and the gallbladder. Briefly the gallbladder, but mostly the liver and the small intestine. We left off last class on Tuesday with this slide where we discussed the production of bile. We had briefly outlined the pancreas and how it's connected to the common hepatic duct and releases enzymes in this now alkaline solution into the duodenum to help break down food, neutralize the chyme as it leaves the stomach, and help break down some of those nutrients. So this is just a schematic showing you the production and storage of bile. We'll look at the route that bile takes from the liver to the gallbladder a bit more in depth. This is just a nice flow chart with arrows showing you, sure, it's produced. There are right and left hepatic ducts that produce and transport bile. They merge in the common hepatic duct, and then when the sphincter of Oddi is closed, they're stored in the gallbladder, or it is stored in the gallbladder until it's needed and is released into the duodenum. Gallbladder is really only concerned with storage of bile. The liver has many more functions. Many more functions, as outlined here. And we're sort of skipping a few steps. If we follow food through the digestive system, we've glossed over the breakdown and the absorption for right now, and we're jumping ahead to the liver. Physically, in the order as we progress down through the body, the liver is apparent and obvious, very near the stomach. But in the route that food takes, we've skipped a couple steps to get to the liver. But I think it's important to outline a few of those functions. And first and foremost, storage of carbohydrates, processing of fat, absorption and elimination of toxins. Liver gets first crack at everything that comes out of the digestive system. And that's why I have the figure shown up here on the right. You remember a slide like this from when we did the in-class activity on venous circulation, right? Remember the hepatic portal vein? We even covered this in class. Special case circulation in the body. Pulmonary circulation was one, and hepatic portal circulation was the other. Hepatic portal circulation drains all of the intestines, all that nutrient-rich blood, mainly coming here through the superior mesenteric vein, which merges with the splenic vein to make the hepatic portal vein and deliver all that good nutrient-rich stuff to the liver for processing. And these are the list of actions that the liver will perform on that incoming blood. Make bile, break down heme and red blood cells, destroy any toxins, activate vitamin D, big role in carbohydrate storage, lipid processing, protein metabolism, and getting rid of unwanted toxins. Now at this point, I think we're comfortable understanding, sure, all the intestines are drained through these branches that merge in the hepatic portal vein. Then something happens. You see it diffuse into the right lobe of the liver. Question mark. Hepatic veins back to the inferior vena cava. This list on the left-hand side is the question mark right now. We're not sure how it's organized or structured, but we'll get into that in a couple slides. Some things happen, blood flows, and then drainage from the liver returns to the inferior vena cava via the hepatic portal veins. Also tucked away under here, notice the gallbladder, which is the storage place for bile, which flows in the opposite direction of the blood moving through the liver. And you'll see that coming up. Now we mentioned some things about the liver so far. What's obvious to look at, or what's obvious to notice about the liver in this orientation? Two lobes. And those of you at Dal saw this plainly. Two lobes of the liver. Right is the largest, left lobe, both separated by the falciform ligament, which I pointed out on the slide earlier in the section. It's a fold of peritoneum. It divides and suspends the liver right underneath the diaphragm. Not functionally, the left and right lobes have very similar roles. They're made of the same base units, functional base units, but structurally divided into left and right lobes. If we zoomed in, so let's take a chunk out of the liver tissue. Here there's a square cut out of the right lobe of the liver, and we're zooming in a fair bit. We've mentioned previously that blood is delivered from the hepatic portal vein to sinusoids in the liver, then to the hepatic veins, returned to the inferior vena cava and back to the heart. But we don't know what the sinusoids are right now. We're going to zoom in and take a look at what the sinusoids are. Jumping ahead of myself. So I've taken a big chunk out of the liver, I've gotten rid of most of it, and I'm zooming in really close to look at some of the cells, the hepatocytes, and the blood flow between, weaving around those hepatocytes within the liver. Now, some obvious notable characteristics in this slide are, first and foremost, we have a few groupings of vessels at the corners around this hexagon. And the grouping of vessels you'll see spread out throughout the liver. It's always a grouping of 3 vessels, and they're spaced out pretty regularly. Pretty regularly. This is the portal triad that contains, you can see a closer version over here on the right-hand side, that contains bile duct, hepatic portal vein branches, and then branches of the hepatic artery, which supplies oxygenated blood to the liver tissue. So these 3 vessels always travel together in the liver. They serve different functions, they're carrying different fluids, blood in different states, but they always travel together. And in this medium picture, you can see they're arranged nicely around this central divot, this central depression, which is the central vein in this lobe or in this section of liver tissue. And the central vein collects everything after the liver's processed. All the blood that has been processed collects in the central vein, and it's the many central veins scattered throughout the liver that combine to drain blood back to the inferior vena cava. So these are numerous sections within the liver. We're looking at one candidate section here to get a sense of the major structures, but there are millions of these in the liver proper. Millions of portal triads, millions of central veins, and we're going to look at their roles in a little bit more detail. So you can see most of the features labeled here. We'll go into a bit more detail as we zoom in on this rightmost figure on the next slide. Portal triad, central vein. Each of the individual squares is a hepatocyte, a liver cell. 80% of the mass of the liver is these cells. The rest will be blood and vasculature, some vessels, the sinusoids themselves, but 80% are these individual cells. Now you can tell it's a very ordered, regular structure, but on close inspection, the structure's kind of odd, right? The structure is kind of odd. We have these hepatocytes that are arranged in what look like strands. You section the liver, you look down on the section, and it looks like they're in strands. And each hepatocyte is connected to a neighboring hepatocyte in these long individual strands that might merge or diverge. And the space between them, well, not yet. They look like long strands when you look at them face on from the top, but when you look down, you can see that they connect in sheets. The hepatocytes connect in sheets called lamina. these layers of hepatocytes fold one on top of the other. They're layered, one on top of the other, in lamina. This is one lamina over here. You can see this strand of hepatocytes dives deep into this section. This is all one wall of liver cell. And over here on the right, you see a couple laminae. There's one lamina over here. There's another one separated by this blue structure in the middle. So many interleaving sheets. And this is a really important organization strategy in the liver. What it does is it creates a space between these sheets for blood to flow so that blood can come into contact with every single cell. Every hepatocyte has access to the space between these lamina. Right? So this is the space. These blue crevices are the spaces. This hepatocyte, for instance, has access to two. Hepatocytes over here have access to the space between the lamina. They're bathed in whatever's being carried in those crevices, those spaces between the lamina. So what is filling those spaces? What is filling those spaces? The spaces themselves are the sinusoids. Sinusoids. The name kind of implies what their structure might be. These long, undulating, open spaces between lamina. So one sinusoid is shown here. This large open crevice that extends between lamina, that's a sinusoid. This is a sinusoid. That's a sinusoid. All these open spaces are hepatic sinusoids. And it's those spaces that receive the incoming nutrient-rich blood. So how does it get there? However we absorb nutrients, they're delivered to the portal circulation. And that portal circulation branches into these individual branches of the portal vein. This is distributed throughout the liver to perfuse the entire tissue, the first element of that triad that we saw on the last slide. So everything in this branch of the hepatic portal vein is nutrient-rich blood coming from the intestines, coming from the superior mesenteric vein. And so nutrient-rich blood travels through this branch. It travels into the hepatic sinusoid, where it's met with oxygenated blood from the hepatic artery. So liver tissue, like any tissue in the body, needs oxygen delivered so that those cells can survive. It needs nutrients delivered so that the cells have energy to do the jobs they need to do. Liver cells need to process incoming nutrients, incoming toxins, and it still needs a supply of oxygen and nutrients to do that. So, blood from the hepatic artery mixes with nutrient-rich blood from the hepatic portal veins, creating this oxygen-rich, nutrient-rich, potentially toxin-rich, bacteria-rich blood that's never seen in other tissue in the body before. And this is the substance that is being processed by the hepatocytes. This mixture of oxygenated blood and nutrient-rich blood from the intestines. So nutrient-rich blood mixes with oxygenated blood in the sinusoids, forming this odd mixture. And all of the sinusoids converge. I have this mixture that these hepatocytes are working on. I have another sinusoid, or three other sinusoids down to the left, with similar blood flow. They're seeing similar nutrient-rich blood from the intestines. They're seeing similar oxygen-rich blood from the arterial system. These pass through the sinusoids to the central vein where everything pools together and blood eventually returns to the heart. Through the hepatic veins to the inferior vena cava back to the heart. So in all of this, every single hepatocyte has been exposed to blood from the intestines, nutrient-rich blood with newly absorbed food, vitamins, minerals, bacteria, alcohol, drugs, etc. Every single cell in the liver. What we're missing in this description so far is bile production. And you can see the green squiggles on the top, the bile ducts shown here, and the canaliculi, these smaller ducts that end up merging with the main bile ducts between hepatocytes. Another job of liver tissue is to produce bile. Notice the opposing flow. Yes, the bile duct over on the right-hand side is part of the portal triad. The bile flows out of the liver in the opposite direction of the incoming blood to be collected in the hepatic ducts and stored by the gallbladder. All three of these things together, again, the portal triad. So every hepatocyte has an easy crack at the incoming blood. These hepatocytes produce bile that's distributed back to the gallbladder and a washing over of nutrient-rich blood through the liver to be processed and returned to the rest of the body. We have some help. What I didn't really articulate on the last slide, but you can see here shown intermittently in these sinusoids are these stellate reticuloendothelial cells. Reticuloendothelial cells that serve to help break down any old, worn-out white blood cells or red blood cells. These help to initiate the bilirubin breakdown and excretion. Any old, worn-out white or red blood cells in the body, and they help to get rid of some bacteria that come in as well. These break down incoming particles to digest those and return those ingredients back to the liver. What I want you to take away from this is the organization of the liver cells and the flow of blood across the hepatocytes through the sinusoids to be collected in the central vein. So we have a very close-up view of the liver tissue here. And it's easy to get lost in this repeating sequence of structures. And it's up for debate. There's still some controversy over what the functional unit of the liver should be. The hepatocytes are the individual cells, but they work in conjunction with the portal vein bringing nutrient-rich blood, the hepatic arteries giving oxygen-rich blood, and then the central veins that return everything back to the circulation. The hepatocyte isn't really the base unit of function in the liver. It's somehow the combination of all of these characteristics. It is the base functional unit in the liver. And for the longest time, the hepatic lobule or lobule was how we thought about the organized structure in the liver. And that's what we depicted on that first slide. That hexagon shape, central vein is central. It makes sense, right? 3 portal triads equally spaced around the periphery. And blood washes over the hepatic lobule inwards to the central vein, where all those functions of the liver are being processed or being accomplished. This model really emphasizes the nutrient absorption role of the liver. Oxygen, nutrients washing from the periphery inwards so the liver can absorb and then dispose of any other byproducts through the central vein. And this schematic was well adopted because this histology is fairly apparent in other organisms, namely in pigs when we're trying to really understand the histology of liver. And we see this organization in humans, but it's not as regular. It's not as much of a regular honeycomb ordered structure, series of structures. So we can pick out these structures, but they're not always necessarily organized like this. And so even though we've classically thought of the base functional unit of the liver as a hepatic lobule, we're starting to change our perception a little bit. The other way that we could think about it would be as a portal lobule, where the central veins define the border of the functional unit. This isn't really widely accepted. This mostly emphasizes the bile-producing actions of the liver. Portal triads in the middle, incoming nutrient-rich blood, but outgoing bile. Not really well agreed upon. But we have a bit more agreement for this third option. The acinar model, the hepatic acinus, as the functional unit of the liver. And what that describes is this oval shape that's highlighted. It's an imaginary oval shape that spans the space between two central veins. And a zoomed in version is shown here. We can see some very familiar structures, the hepatocytes ordered in layers with the sinusoids between them. And the main branches of the portal triad in the middle, a highly vascularized region in the middle with outward flow of blood towards the central veins on either end. And it's hard to see defined borders if you were to look at tissue in the liver, but you can imagine what it would look like if you found two central veins. You would divide these arbitrarily by long and short axes. The short axis follows the length of the portal triad veins. The major axis is the short axis in the middle. And then these imaginary arcing lines on top and on the bottom forming the long axis of the hepatic acinus. Now here they're nicely shown. The hepatocytes, the lamina, organized in these nice curved structures. But they're not always like that in practice in the tissue proper. So you have to imagine this nice arcing curved structure. But the order of these structures would be the same. Major circulatory provision in the middle at the portal triad, and then two central veins bordering the hepatic acinus for return of blood back to the heart. This is the preferred structural and functional unit of the liver in recent years. It includes all of the elements that we've described. It helps to describe flow of blood through the liver tissue from this point of entry at the triad to where it exits at the central vein. So the structural elements are there. And there are functional elements too, and I'll describe zone 1, zone 2, zone 3 shortly. But this supports our observations of how sometimes glycogen, for instance, is clustered in little areas within the liver. Or fatty liver disease. Fat tends to accumulate in little pockets of the liver, and it almost ends up looking speckled. And thinking back to the cadavers we saw at Dow, there was a really good example of that, where the liver had this uniform speckling all over the surface, and it could be the deposition of fat in these individual locations based on this model of liver function. So, how does this model help describe the function of the liver? If we generate this picture with the arbitrary long and short axes, we also create what would be theoretical zones. Zone 1, Zone 2, Zone 3, with Zone 1 being closest to blood supply, Zone 3 being furthest away from blood supply. Zone 2 being in the middle, but that goes without saying. I'll describe Zone 1 and Zone 3 And then to figure out what zone 2 is, take those two things and cut them in half. So zone 1 describes all of the liver cells that are in immediate proximity to new blood supply. These are the first cells that will be exposed to incoming glucose. So these are likely the cells that will take up and store glycogen. They will probably have more glycogen than cells in the other zones. These are also the cells that will be exposed to toxins or damaging substances first. They're the first to degrade in response to abuse, overconsumption of alcohol, for instance. And we see little pockets of cells that degrade first near the portal triad. They're also the first to regenerate. So if you are a recovering alcoholic and you've done damage to your liver, the cells in close supply to the arterial blood are the first to regenerate. And we notice that as well. So these cells in zone 1 are very responsive to whatever is being delivered to them. Healthy vitamins and minerals, glycogen, they'll store it. Toxins, they're damaged, but they always have this ready blood supply. Cells in zone 3 are the opposite. These are the first ones to be damaged. If there's ever a lack of circulation in the liver, cells in zone 3 are the most susceptible to that lack of blood supply. These are also the cells that tend to store fat the most. During fatty liver disease, steatosis is the technical name for it. These little pockets near and bordering the central veins take up what's left over. And a lot of the time, fat is left over that can be absorbed by these cells. And we see these pockets, the speckling of regions with a high fat content with fatty liver disease. And then zone 2 is somewhere in the middle. Somewhat susceptible, somewhat able to store glucose, somewhat able to respond to toxins, but somewhere in the middle. So we have this zone close to circulation, zone far away from circulation, and they result in the phenotypes that we observe at the tissue level. So we're starting to consider this as the main model of structure and function in the liver, not just on an individual cellular basis. So we've looked at the detailed flow of blood through the liver. Nutrient-rich portal blood mixes with oxygen-rich arterial blood in the sinusoids that expose all cells of the liver to whatever is being taken in by the body. That portal blood will have glucose, will have some fat, it will have ions, minerals, toxins, etc., some drugs, microbes, bacteria, anything that you're taking into your body that you are ingesting that goes through the digestive system. In the sinusoids, those two fractions mix, and this is the site where oxygen is extracted that supports normal liver cell function. Normal liver cell function is to absorb nutrients, extract toxins, produce bile, etc. And so the incoming arterial blood supports the actions of those liver cells on the incoming portal circulation blood. And then as waste products are deposited, or not only waste products, but any elements or substrates that are needed by the rest of the body, if glucose is required, the liver will release glucose. As fat is repackaged into cholesterol, the liver will release fat. Those are released, any newly formed substances are released into the circulation from the sinusoids into the central vein, and then down this series of steps towards the heart and back to the rest of the body. It's a really elegant design, I think. The liver is like a sponge. Fluid washes over every single cell with this unique layering of the lamina, and then the drainage through the central vein. Really, really interesting. We haven't even touched on any endocrine or cytokine signals, hormonal signals at the liver. Any questions about the organization of these cells? The organization of the structures and blood flow through the liver? So far, so good. All right, let's introduce the small intestine. Might have another 5-10 minutes in class. We're picking up with food as it travels through the digestive system. So we've Tarantino'd it. We've jumped to the end, the liver being where all the nutrients are absorbed and processed and released. This is where they would have been supplied. This is where the liver is supplied from. Food in the small intestine that's digested and absorbed. And this is by far the most important site for digestion and absorption in the digestive system. Massive surface area owing just to its length. It's 3 meters long, the small intestine. And you've seen some of the characteristics in lab that help to boost that surface area. 3-meter tube has a lot of internal surface area, but when you start to add folds and villi, finger-like projections, and microvilli on top of those finger-like projections, all of those characteristics tend to increase the surface area, which are the sites for digestion and absorption. And we'll touch back on those coming up. The major site for digestion and absorption in the body. Small intestine made of 3 parts, like you know. Duodenum, jejunum, ileum. In that order. Duodenum being the smallest, the shortest section, about 20 centimeters, 25 centimeters in length. Jejunum being 1 meter, and the ileum being 2 meters in length. Very, very long organ or part of the digestive system, this is. I always remember, I've told a few people in the Monday afternoon lab section, I always remember the order as that company that makes those drones, DJI, make Phantom, Phantom 2. That's how I remember the order of the duodenum, jejunum, ileum. But if you don't remember DJI, if you're not a big drone fan, duodenum we know is where the food is released from the stomach and the bile is secreted. Jejunum, top left quadrant. Ileum, bottom right quadrant. And there's really very little overlap. They're generally quite separated in that regard. Now, to trigger your memory from the arterial activity that we did in class, the cells of the small intestine also need a ready supply of oxygen and nutrients to do their job. They don't get it from the food that's coming in through the digestive system. They need a supply of oxygenated blood, which comes through, mainly the superior mesenteric artery. And you can see branches of the jejunum in the upper left quadrant, branches of the ileum in the bottom right quadrant. The major part of the small intestine has been cut out to give access to and so we can view the arterial branches of the superior mesenteric artery. But you can see they're pervasive. They're all over the place in the mesenteric. And these are the branches of the arteries that are contained within the folds of the peritoneum. If you were to open someone up and you looked at their small intestines and you picked them up, they're connected. There's flaps of peritoneum that wrap around the small intestines containing all of these individual arteries and veins, supply from the superior mesenteric artery and then drainage back to the superior mesenteric vein. So the small intestine, the major site of absorption and digestion in the body. What does it do to help absorb and digest food in the body? Like with the esophagus, we have motor actions in the small intestine. Peristalsis, contraction, segmentation is new here, but churning and mixing of chyme from the stomach with pancreatic juice from the pancreas, and now also some intestinal juice to create this really nice fluid full of nutrients that can be pressed into service to help absorb into the body. So we have segmentation, peristalsis, function of the digestive system to move that food, churn that food, mix that food. Secondary action is after mixing and churning that food with the surface of the small intestine in close proximity, that foodstuff can be broken down and absorbed. More complex carbohydrates, proteins, nucleic acids, fats are broken down and absorbed, 90% of which are absorbed in the small intestine, only a small fraction ever absorbed in the stomach, smaller than small fraction in the mouth, and very little in the large intestine, which is coming up, but some ions, some salts, some fluid in the large intestine. Now, being that this is a list of the major functions of the small intestine, the characteristics here that are unique, that support these functions, are those that we just mentioned. We need a big surface area, we need some muscular activation, and the surface area is achieved through many circular folds, the villi, which are finger-like projections, also known as tufts of hair. Villi translates loosely as tufts of hair. And then microvilli, which are small tufts of hair. Projections from the walls of the inside of the small intestines that increase surface area, increase the opportunity for absorption. Circular folds, you can see on this slide, they are obvious and visible to the naked eye. They might extend into the lumen a centimeter in some cases. Some of these folds will encircle the entire lumen, all the way around the small intestine. A giant ridge blocking the flow of food through the small intestine. Some only cover part of the inside of the lumen. And you can see if we spread out the small intestine, lay it flat on the table here, the undulations in these ridges. Other than increasing the surface area, there's obviously more tissue, there's more surface here than if this were just flat. These serve a second purpose of making it hard for food to travel. These block the flow of chyme in the small intestine. And they don't block it completely, they just block portions of it or a side of the small intestine. And so the chyme is forced to spin and move and spiral, to mix, so that we have uniform access of enzymes to foodstuff, and so that we have uniform access of the food in the small intestines to the wall of the small intestines, which we need because the wall is the site where we absorb food or absorb nutrients from the food in the small intestines. So these folds are integral in increasing surface area, but also making resistance to flow, helping to churn food as it moves through the small intestine. We'll talk about movement through mechanical digestion, and then we'll call it for the day. So as food moves through, like the esophagus, we have contractions that propel chyme through the small intestine, that mix and churn and bring it into contact with the walls of the small intestine. Like the esophagus, we have circular muscles that go all the way around that can pinch off a section of the small intestine. And we have longitudinal muscles, like pulling on the sleeve of your jacket, that can shrink and expand lengthwise sections of the small intestine. Now, the circular muscles are largely used in segmentation. Segmentation is a process that describes, think of it as the chopping up of food. Alternating contraction and relaxation of neighboring regions in the small intestine. The book finally caught up with the analogies. They say this is the same as trying to pinch a closed tube of toothpaste. So hold a tube of toothpaste and pinch it and relax it back and forth, back and forth. That's segmentation. And so the circular muscles pinch off, not completely off, but compress food, mix the chyme, break the contents up. And this is mainly active in situations or locations where the small intestine is distended. It makes sense. We know there's food there, so let's segment it and break it up into smaller and smaller pieces. That continues until distension wears off. When that chyme has been broken up, when those particles have been broken up sufficiently, there's less distension, and we have peristalsis that activates, that propels food forward. So in segmentation, there's no propulsion. There's no forward movement of the food. It's generally static. It's breaking up of that food at that location only. And once that food's broken up, peristalsis activates. The longitudinal muscles activate to push food through the small intestine. Like the intestines, like the esophagus, they're wave-like contractions. But it's slightly different in the small intestine. It's a very long organ, 3 meters long, versus your esophagus that might have been 30 centimeters. And so you can imagine peristalsis here as 5 esophagi lined up together. Think of it that way. And what I'm describing is what's called the migrating motility complex. So you'll have peristalsis in one region, that stops. Peristalsis in one region, that stops. Then peristalsis in another region happens, and then it stops. And you have this sequential series of peristaltic contractions that end up pushing food all the way through the small intestine. In total, it would last 2 hours, whereas the esophagus peristalsis completed in 20 seconds, 5 seconds. So let's leave it there. We will not pick this up until next week, which is actually perfect because the next big section is digestion and absorption, which is fairly large. And we got some nice diagrams and schematics and pathways. If you have any last-minute questions, please don't hesitate to reach out and email. Good luck studying tonight. And I'll see you at 1 o'clock tomorrow for our midterm.